Concept 17.4, Translation, a closer look. In this concept, we will be talking about the molecular components of translation, building a polypeptide, and completing and targeting the functional protein. Remember, translation is converting the information in an mRNA to a protein. To do this, you need an mRNA, or RNA copy of a transcription unit. You need a ribosome, which is an organelle that is composed of rRNA and proteins. Ribosomes come in two parts, the small subunit and the large subunit. Another type of RNA, the tRNA, brings in amino acids to the ribosome. The ribosome hooks these amino acids together to form a protein. So, how does this all happen? Let's start with the tRNAs. Each tRNA has a specific enzyme or amino-acetyl tRNA synthase. The synthase has an active site that will only allow one type of tRNA and one type of amino acid in. It welds them together, and the tRNA is now an amino-acetyl tRNA, or a charged tRNA. This is how the tRNA knows, so to speak, which amino acid it should be carrying. Let's look at the ribosome. The large subunit has three sites. The A site is where the charged tRNAs come in. The P site is where the amino acids are hooked together. The E site is where the empty tRNAs leave to go get another amino acid. The small subunit has a site for binding the mRNA. So the small subunit binds to the mRNA. Then the first charged tRNA matches up with the first codon. The large subunit clamps down over the tRNA and the mRNA. This is the end of initiation of translation. The ribosome chugs down the mRNA with the charged tRNAs coming into the A site. The growing peptide chain is moved on to the new amino acid, and when the empty tRNA ends up in the E site, it leaves to go get another amino acid. Let's watch an animation that shows this very well.
In prokaryotic cells, translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formal methionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA-carrying amino acids can bind. One is called the peptidyl, or P site, and the other is called the acceptor, or A site. There is also a third site called the exit, or E site, where transfer RNAs are released. The initiating transfer RNA carrying formal methionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formal methionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of one codon, and the transfer RNA that carried the formal methionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of one codon. As the shift occurs, the two amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
feel free to watch the preceding animation all you like. Make sure you study the textbook as well. Both are important. In eukaryotes, at the end of the protein is a signal peptide. And as it comes out of the large subunit of the ribosome, a signal recognition particle, or SRP, attaches. The SRP moves the whole bundle to the rough ER. As the protein is formed, it is folded into the proper shape. Once it is done, the signal peptide is removed, and the protein is now in the lumen of the rough ER and is ready to go to the Golgi. Cool, huh? Well, that's it for this concept. Once again, make sure you write down questions that you may have.